Dear viewers, greetings. In this video, we are going to see about ultraviolet spectroscopy. In short form, it is called as UV spectroscopy. This video will contain the following contents. First, an introduction about ultraviolet spectroscopy, followed by principle of UV spectroscopy, instrumentation of UV spectroscopy, steps of UV spectroscopy, applications of UV spectroscopy, advantages of UV spectroscopy, and finally limitations of UV spectroscopy. Ultraviolet spectroscopy. Ultraviolet spectroscopy or UV spectroscopy is an analytical technique that measures the absorption of ultraviolet light by a chemical substance and providing information about its electronic structure and concentration. UV spectroscopy operates typically in a wavelength of 100 nanometer to 400 nanometer, focusing on the interaction of UV light with matters. UV spectroscopy is a valuable tool in various fields, including chemistry, biochemistry, microbiology, biotechnology, and environmental science. Principle of UV spectroscopy. The principle of UV spectroscopy is based on the interaction of UV light with molecules. When UV light is absorbed by a molecule, it excites the electrons to higher energy levels. The wavelength of UV light that is absorbed depends on the structure of the molecule. By measuring the absorbance of UV light at different wavelengths, we can obtain information about the molecular structure and concentration. Instrumentation of UV spectroscopy. The UV spectroscopy contains 5 major parts. They are light source, monochromator, sample holder or cuvette, detector, and finally display or output device. Light source. Light source provides UV light, usually from a deuterium lamp, and for UV region the range is 160 nanometer to 370 nanometer, and sometimes a tungsten lamp for visible region, and its wavelength ranging from 375 nanometer to 800 nanometer. Second, monochromator. The monochromator disperses light into its component wavelengths and allowing selection of the narrow band of wavelengths to pass through the sample. Third, sample holder or cuvette. The sample is placed in a quartz cuvette as quartz is transparent to UV light. The detector measures the intensity of light which is passing through the sample and converts it into an electrical signal. The photomultiplier tube or photodiode array detector is commonly used. Finally, display or output device. The data, particularly absorbance versus wavelength, is displayed graphically or numerically for the further analysis. Steps in ultraviolet spectroscopy. There are 5 steps. The first step is sample preparation. The sample, usually a liquid solution, is prepared, ensuring it is transparent to UV light in the region of interest. If necessary, dilutions are made to ensure that the absorbance is within the instrument's linear range. The second step is selection of wavelength. A specific wavelength of UV light is selected based on the type of electronic transition which is expected in the compound being analyzed. The third step is passing ultraviolet light. UV light is passed to the sample in the spectrophotometer and the detector measures how much light is absorbed at each wavelength. The fourth step is measurement of absorbance. The amount of UV light absorbed by the sample at different wavelengths is recorded. This produces an absorption spectrum and showing absorbance or transmittance versus wavelength. Finally, the step 5 is data analysis. From the absorption spectrum, the wavelength corresponding to maximum absorption is identified. Using Beer-Lambert's law, the concentration of the sample can be calculated if the molar absorptivity is known. Applications of ultraviolet spectroscopy. The first application is quantitative analysis. The ultraviolet spectroscopy is used to determine the concentration of compounds that absorb UV light such as proteins, nucleic acids and organic molecules. The second application is qualitative analysis. In qualitative analysis, UV spectroscopy is very helpful for identification of compounds based on their absorption spectra, particularly in the study of conjugated systems. In pharmaceuticals, UV spectroscopy is used in quality control to ensure the purity and concentration of drugs by comparing their absorbance at specific wavelengths. In biochemical analysis, ultraviolet spectroscopy is used to study the biomolecules such as nucleic acids and proteins which have characteristic absorption in the UV range. For example, DNA absorbs around 260 nanometer. In environmental monitoring, UV spectroscopy is very helpful in detection and quantification of pollutants such as nitrogen dioxide or organic compounds in water. Finally, in structural studies, UV spectroscopy supports in determining the presence of conjugated double bonds and aromatic systems which absorbs UV light at specific wavelengths. Advantages of ultraviolet spectroscopy. Ultraviolet spectroscopy is a versatile technique with several advantages. Here, there are some key benefits or key advantages. The first one is sensitivity. UV spectroscopy can detect even small amount of analytes, making it suitable for trace analysis. Next, specificity. UV spectroscopy can be highly specific for certain compounds, allowing for their identification and quantification without interference from other components in a mixer. Simplicity. UV spectroscopy technique is relatively very simple to perform and requires minimum sample preparation. Speed. UV spectroscopy measurements can be made quickly and making UV spectroscopy suitable for many routine analysis. The next advantage is non-destructive. UV spectroscopy does not destroy the samples and allowing for further analysis. Wide applicability. UV spectroscopy can be used to analyze a wide range of compounds including organic substances and inorganic substances. Qualitative analysis. In qualitative analysis, UV spectroscopy can be used to determine the concentration of a substance in a solution using the Beer-Lambert's law. Finally, qualitative analysis. In qualitative analysis, UV spectroscopy can be used to identify the presence of specific functional groups in a molecule based on its absorption spectrum. Overall, ultraviolet spectroscopy is a valuable tool with many advantages that make it a popular choice for a variety of applications. Finally, limitations of ultraviolet spectroscopy. Ultraviolet spectroscopy has some limitations. The first limitation is limited wavelength range. UV spectroscopy is typically limited to the ultraviolet and visible regions of the electromagnetic spectrum. This can be a limitation for analyzing compounds that do not observe in this region. The second limitation is interference. The presence of other absorbing species in a sample can interfere with the analysis and making it difficult to accurately quantify the target analyte. The third limitation is lack of specificity. While UV spectroscopy can be specific for certain compounds, it may not be able to distinguish between isomers or closely related compounds with similar absorption spectra. The fourth limitation is sensitivity limitations. For some analytes, the sensitivity of UV spectroscopy may not be sufficient for trace analysis. The fifth limitation is matrix effects. The presence of other components in a sample can affect the absorption properties of the analyte and leading to errors in quantification. The sixth limitation is Sample preparation. Sample preparation can be time consuming and may require special techniques, especially for solid or heterogeneous samples. Finally, the seventh limitation is interference with solvents. The solvent used to dissolve the sample can also absorb UV light and potentially interfering with the analysis. It is important to be aware of these limitations when considering the use of UV spectroscopy for a particular application. In some cases, other analytical techniques may be more suitable or can be used in combination with UV spectroscopy to overcome its limitations. Dear viewers, that's all about the ultraviolet spectroscopy. Thank you for your support. Thank you.